Our last speaker of the morning is Dana Pe'er. She grew up in Jerusalem, where she got schooling at her 3 degrees at the Hebrew University in Jerusalem. She then went and did a postdoc with George Church at Harvard Medical School in the Department of Genetics. And then she went to Columbia, where she began to use her computational skills to begin to understand biological systems. Now, there's an apocryphal story, which I suspect is true, that she actually had a head start, because it is said that when she was in 2nd grade, her father showed her mathematical proof that the number of irrational numbers greatly exceeds the number of rational numbers in the universe. Is that correct, Dana?
Yes.
So life is unfair, because most of us did not have fathers who in 2nd grade challenged us with complex mathematical theorems. You're close to being ready?
I don't know, this thing isn't,
We have the dreaded PowerPoint pause.
There. Okay,
We're there? Good.
so thank you very much for the kind introduction. Is it,
What?
it's not screening up here. Ah, it is, okay. Thank you very much for the kind introduction and for inviting me here to this wonderful symposium. I've been enjoying every single talk till now. This one will be not the best one. And the topic of this conference is heterogeneity within a tumor. And often when I speak, I have to convince people why we want to do it. Certainly not in this audience, but I do want to convince you of something else. Most of the morning, we talked about heterogeneity of the DNA within the tumor. And certainly DNA is the genetic driver of tumors, and there is much to be studied through the DNA. But DNA isn't the entire story. We heard there's epigenetics. And all this comes down to diversity in the RNA, in the protein, in what the cell is phenotypically doing. And we need to begin querying that heterogeneity as well. We actually need to understand the different cell states, the different cell abilities, the different things that cell seem to be doing in the tumor. And I see we have this somehow isn't working nicely. Should I switch to the other one?
Yeah, maybe the cord, something's wrong with that cord.
This is not my cord, but...
There we go, there we go.
Okay, I will hold this cord the entire talk.
That's fine, let it go,
Okay.
let it go.
And so I want to describe what I think is cell state and also explain why Gary's making all this effort to try and increase dimensionality. So cell phenotype, often we look at these bivariate plots. You can imagine this is CD34 and CD38 talking about some stem cells, but you can think of a cellular phenotype as what a particular cell is expressing, either genes or proteins, and you have a multi-dimensional phenotype, and you seem to have here 2 cell phenotypes, one that's high in protein A and one that's high in protein Y. But if we measure another dimension, suddenly it's revealed that there's actually 4 clusters here, 4 phenotypes. We just didn't see that missing protein Z. And as you go higher and higher, you can distinguish more and more phenotypes, more and more combinations of possible genes and proteins and their behaviors and what they actually mean. But the problem is that as we increase the dimensions, we love looking at 2 dimensions. We can sort of handle 3 in this picture right here. But when we get to 50 dimensions, 300 dimensions that we heard in the previous talk, and then 20,000 and more dimensions that you get with RNA-seq, then you have a problem of, oh God, how do you interpret this data? How do you even visualize this data? We as human beings aren't very good at going beyond 3 dimension. So the goal is to take the type of data that my magician friend is creating for me, making me feel like a kid in a candy shop, and actually figuring out how to analyze it. And the first thing we like to do as humans is visualize. And dimensionality reduction, taking high dimensional data and trying to push it back to meaningful 2 dimensions is what we've been doing for a long time. This is linear dimensionality reduction, so we basically do PCA. And this is bone marrow. We have markers to identify different immune subtypes, and these are identified here. Every dot here is a single cell colored by manual gating and classification. And you see that linear the reduction doesn't quite cut it. So we developed VSNE. It was based on TSNE by Mateen and Hinton, which is basically trying to take a nonlinear dimensionality reduction. Again, every dot you see here is an individual cell, and they're plotted based on their relative similarities and dissimilarities. So in between every single pair of cells in this image, their relative distance, if they're close, that means they're very similar. If they're far, they're very distinct. And it actually automatically identifies, without knowing anything about the immune system, the different cell subtypes, the T cells, the monocytes, the B cells. And again, this is visualizing multiple dimensions in 2 dimensions. If I sort of screen it down here, these cells right here are these blue cells here in 2 dimensions, but in one visual, we're capturing the similarity and dissimilarity in all dimensions. We can do the same thing for 3 dimensions, and this is the same data as before. I've been showing you the same bone marrow data, now projected down to 3 dimensions. This is 13 down to 3, and you see that immune subsets are not these nice round convex ellipsoid balls, but they actually create very complex high-dimensional concave twisty and turny shapes that actually also reveal a lot of the regulatory forces that regulate them. And I want you to remember this cup and ball, which we see in this picture, because this cup and ball is pervasive in cellular phenotypic space. So I want to tell you one story of how you take the wonderful technology developed by Gary, combine it with a little bit of computational methods, and try and really drive in to understand something about cancer heterogeneity, in this case, a pediatric AML cohort. This is, of course, joint work with my partner Gary, and Jacob, a grad student in my lab, and Aaron, who was at the time a grad student in Gary's lab. So we took an AML cohort as well as a couple of healthy normals. I think it's very, very important to always compare to normal, so you know what the normal and the abnormal is. And you can ask, what are the phenotypes present? How do they differ between patients? How many phenotypes does a single patient have? Are these clinically significant? What is the phenotypic landscape? And the idea is, again, to take these patients and measure them with a whole bunch of markers, both surface markers to say, who am I, what type of immune cell I am, and what am I doing, some signaling markers that have been perturbed and interrogated, as Gary mentioned. But this was actually a pretty challenging data set. So we had 16 million individual cells measured in 31 dimensions. And as I showed you, these clusters, they're not nice looking, they're not convex or even ellipsoid or anything like that. Most clustering algorithms fail, and they change in orders of magnitude. You have many less stem cells than you have CD4 cells or blasts or anything else. And therefore, we really needed to develop a new clustering method for this type of data that could really resolve it. Again, since we don't know cancer, since we don't know what's working and not working, then we need to first study and benchmark and feel confident with a system where we do know the answer. So again, we started with healthy bone marrow, and here's 500 cells randomly sampled from healthy bone marrow, organized in this map where every single node is a cell, and every cell is now connected to its neighborhood of most similar cells. So this is like building a social network for cells. Every cell build a community of similar cells in a graph-like structure, and the topology of this graph is going to help us reveal the cell subtypes. So as you can see, the structure of this graph is very modular, and every one of these modules right here, it's CD34 high cells. They're the primitive progenitor cells. The key to making this general idea working is building this graph. You want to build this graph so it looks this nice, so you can, by eye, see how nice and easy it is to split up. And the key here is to develop a good distance metric. And again, a naive metrics, almost any geometric distance metric, here I'm going to show you Euclid, but pretty much everyone fails, is that because of this cup and ball situation that we have pervasive in our space, even when you do k equals 2, connect these cells to their nearest neighbors, suddenly you sort of take this ball and connect it to the cup. It gets worse and worse as you have higher and higher k's, and basically you get connections of often small, important phenotypes, and they sort of get engulfed completely by some of these bigger phenotypes. So what we did is we actually used the jacquard method, which we heard earlier today. and basically said the similarity is not their similarity in their phenotypic space and how similarly or differently they express CD3, but in how many neighbors they share. And so now we build a second iteration of the graph where we fix this and say the distance between you, the similarity between you, is not the direct similarity between you, but the similarity in the number of friends you share. And this thing scales with local density, it removes spurious edges, it really finds tight, small populations, and finally reveals very accurately this phenotypic structure. So now you have a beautiful modular structure driven by a distance metric, and the only thing you need to do is find it. Here it's pretty easy, it's 500 cells, but I was talking about 16 million cells, computing on 16 million cells before people graduate and funds run out is hard. Fortunately, we have the social networking, the internet, the Facebook, and everything else, and they've actually figured out some really rapid algorithms that we can hijack to find the modular structure of the graph once it's correctly built. So again, I want to convince you that this is actually finding phenotypes quite well. So here we have a hand-gated, and here we have our own phenograph algorithm, and as you can see, they match very accurately. This is a hard problem, so I'm giving you the two winners from a recent competition on clustering and gating algorithms for flow, and you see that these algorithms don't do as well. When they work in lower dimensions, they're okay, but as you increase dimensions, some of the known algorithms out there fail. Again, here's another benchmarking to show that as we repeat time and time again, subsample, change your single parameter k, nothing happens, and here's all the competing methods. The higher up you are, the better you are. That means you're more accurate. The tighter you are, that means you're more robust. That means you do it again and again and again on different subsamplings of the graph. And we have confidence on the healthy system, so now we can take this and use it to try and define the phenotypes in the AML cohort and then hopefully believe these phenotypes. So we're going to run our phenograph algorithm, but again, now we have clusters. We have we have exactly 16 patients, and each of these we have about 28 to 35 phenotypes, clusters. And how are we going to, that's about 600, how are we going to visualize all of them? Well, we have that same t-SNE-v-SNE reduction of mapping these clusters based on the similarity now of the clusters and not of the individual cells onto a single map. So this is the AML cohort landscape map. Each color right here represents a different patient. The blue going down here are 5 replicates of healthy cells from healthy donors. And you see that this landscape map is very well mixed. So every region of the map has multiple patients. Every patient is spread out across the map. And if you want to interpret what this map is, it's sort of hard to understand when you look just at patient ID. We can begin looking at what the different markers express in each of these clusters. So you see up here near the healthy progenitor cells, we have a CD34 expressed. Here at the bottom, near the healthy monocytes, we have CD11B and all sorts of different developmental markers in between. And actually, the vertical axis here sort of matches healthy myeloid development. And again, just to give you an idea, remember every dot here is a patient. Now I'm going to show you the same view, but mapping one patient at a time. Here's the first patient, and you see in gray the healthy cells, in yellow the cancer cells. Here's SJ2. SJ2 and SJ1 have the same genetic mutation. Here's another one with a different genetic mutation having more mature cells, more primitive cells. But can we compare these clusters with more rigor? I mean, these are just pictures. They're pretty pictures. They're colorful pictures. Color seems to be the theme that Nellie started earlier this session. Again, we have the phenograph algorithm. So now we're going to do meta-clustering. We're going to take our clusters. We're going to send them and build a phenograph for them, and now do metaclusters. And we found that there's 14 metaclusters for these tumors. And these metaclusters are actually very stable. So when we remove 1 or even 2 patients, we get the same metaclusters. These AML clusters have many different genetic backgrounds. AML is a very genetically heterogeneous disease. Yet again, these different genetically caused AMLs lead to the same palette of 14 phenotypes. And these phenotypes are computationally robust. As we perturb the algorithm, as we remove patients, there seem to be really the 14 metaclusters of AML phenotypic space. Again, we can take these metaclusters and now define every single patient with which metacluster, the palette of the frequency and occurrence of these metaclusters. So we see up here the healthies have the metaclusters that match healthy disease states. And as you go down, every single tumor has its own combination and frequency of metaclusters from this palette of 14. Every color is a different metacluster. But what we do note is when you cluster these, they do cluster based on the driving genetic mutation. Here we have the CBF clusters, the CN clusters, the NMP clusters. These are all basically the genetics drives which phenotypes and in what frequencies you choose. And even with a cohort size of 16, this clustering is very significant statistically. Okay, so we defined 14 metaclusters. We showed that they occur across all AMLs, including the different genetic backgrounds of AML. And we showed that they are driven in terms of occupancy by the genetics. But what do they mean? And up to now, we've only been looking at a surface. How does a subpopulation look? What surface markers is it expressing, but not how does it behave? These surface markers are used to phenotype these in the healthy system, but typically don't have any causal role in the different behaviors of the system, B cell or T cell or whatnot. So we want to query how they behave, and that's what Gary was talking about of interrogating them, asking these cells questions. And we interrogated them with 17 different conditions. So in addition to the surface phenotypes that we have for each of these clusters, we also have a signaling vector for them. That is for each of 16 internal markers, different signaling markers and PSTAT5 and others, how are they responding to different growth factors, stem cell growth factor, IL-7, and giving us a rich phenotype of 224 responses, the response of STAT5 to stem cell growth factor. And we can compute this matrix. Red is high, blue is down relative to the normal basal state. So we have these two objects, and now let's look at both of them. And we see that when we cluster based on the feature, and we selected features that seem to separate healthy and in the healthy primitive cells and advanced cells, we see that there's a couple of very distinct signaling features that basically respond, or there's a signaling footprint for healthy progenitor stem cells. These are all CD34 plus right here. But when we looked at the AMLs, we saw that same exact signaling signature of these 5 signaling responses. But on CD35, there was a complete disruption. Only about half of them were expressing CD34, the other not. So we see a complete disruption of surface and signaling, a very distinct signaling pattern that's conserved with the healthy primitive cells, CD34, completely unconserved, and you can see here this quantitatively, it's about half-half. There's no difference in CD34 expression between the primitive and the mature behaving cells based on their signaling rather than their surface phenotype. We can now do this more systematically. We have 2 phenotypes. We have a surface phenotype. We have a signaling phenotype. And we ask, can we use actually this surface phenotype to classify cell types? The signaling, yes, the signaling to classify cell types, and the answer is yes. So we can take each cluster and look at the surface and say, does it look like a mature or a primitive cell based on its surface? But we can also look at the signaling and say, does it signal like a mature or primitive cell? And our misclassification rate is very low in both cases on healthy. If we know the answer, if we have healthy, we get this extremely accurately. So now we can say, let's define a primitive cell based on its functional behavior rather than its surface expression and see what we get. Here's one patient. Up here on the top are the subpopulations that signal like primitive cells, like primitive healthy cells, and up at the bottom are ones that signal like mature cells. When you look at the surface, you actually see some of these cells within the same patient. This is one single patient, are CD34 plus. Here, every single row, you're looking at a different subpopulation. Every column is either a surface marker here or a signaling marker here. When it's red, it means it's induced. Here, again, red is high. And so we see these CD34 plus populations that signal like stem cells, but within the same patient. These are CD34 low, but are signaling like stem cells. And here, we have CD34 high cells that are signaling like mature cells. Again, a complete disruption. I would like to also say that this particular patient, we actually sorted and based on some of these phenotypes, and both in terms of the surface phenotypes and the signaling phenotypes. This is a multiclonal patient. It has 5 different DNA clones, yet these DNA clones distributed uniformly across these functionally behavioral, epigenetically derived phenotypes. Again, stressing the importance of looking at function and not only DNA. Here's a completely different patient. In this patient, it actually has some CD34 plus cells. None of them are primitive. And in fact, all the primitive behaving cells, based on their signaling response, are CD34 minus cells. And we have such a signature and classification for every single of our patients. And we see that every patient has a number of subpopulations that behave like signaling populations. And these vary in their frequency across patients. And we could look at the gray bars. That's the frequency based on surface. So if I were to look at your surface, how would I predict how much of the fraction of your cells are primitive? And the black bars are based on the signaling. Again, you see that they're not completely agreed upon. Which correlates better with survival? And we actually only had 16, so it wasn't enough. We used these 16 and looked at their frequency to look at frequency in matched gene expression arrays to see what genes correlated with their expression with the frequency of these primitive populations. And this led us a signature that we can now take into different larger cohorts of microarrays. And we actually saw that our signaling-derived signature had very, very, very strong survival prediction. And the surface-based phenotype was not at all significant. So in summary, again, in this case, the leukemia landscape was a number of different phenotypes that were selected from a limited palette that was determined in part by genetics but shared across the different types. That surface and phenotype are completely disrupted in cancer, so using actually surface profiling to try and understand malignancy might not be a good idea. We defined a new clinically, functionally derived signaling phenotype that could bridge the fact that no one could find a single surface marker that matched these. These tools are available. You can use them with any technology, so I gave you one example of how you can actually use this to drill down and find something interesting, but they're all available. They could be applied to different systems, different cancers, as well as different technologies, such as the other technologies Gary talked about, some of the single-cell RNA-seq that's being developed across the road. And I'm giving you a sneak peek. We've been starting to apply it very aggressively to tumor immune microenvironment. And again, this is the value of going in without preconceptions, without trying to gate out the populations you know in advance. This is actually a small flow data set. This is only 8-color flow taken from melanoma patients before and after therapy. And this is one of the responders. And again, we're visualizing it with our VSNA visualization. Blue is before therapy, red is after therapy. We also have phenograph clusters for this, and we see that the signal population, which is very, very clear right here, that changes most in its size, that grows 9-fold following therapy, is this population right here. We see a similar population in a number of other patients that respond to therapy. This is melanoma. And no other population grows more. So what is this population? And we started looking at it, and it's not a phenotype of any known immune type. In fact, it's defined by a couple mid-level markers, and no one would have looked for this. And even if you looked exhaustively, it's actually defined by mid-level expression, again showing why you have to look in a data-driven, unbiased manner with multi-dimension, and now we're increasing this to 17 and 40 dimensions and exploring it more systematically on bigger cohorts. And finally, I want to tell you a little story, and this is going to be very short because I don't have much time left, but I want to give you a flavor of how far you can go. Now that you have single-cell data with multiple dimension, you can also learn from them. You could look at the stochasticity and the variability of the different markers you measured and actually learn networks. So how would you do that? Well, if protein A influences protein B, if protein B or phosphoprotein B is a function of protein A, you expect to be able to see this dependency in a plot that compares these. And now we can use each cell as a point to actually learn these functions. And that's exactly what the Drevey algorithm that we developed does. How does protein A influence protein B? And also you can see, does it influence protein B? Is there a significant relationship between them? Is there a connection? So again, this is work by a postdoc in my lab, Smita, a graduate student in Bern Bod-Miller's lab in Zurich, Nevena, and Bod and Miller. And we want to look at AMT, and I thought it was very important to talk about EMT here in this setting. And I also wanted to stress that even in a cell line, you should look at heterogeneity. And here we're actually trying to drive the EMT using TGF-beta. And this is these cells after a few days. And you see that even at day 3, right here, you have a mixture. This is a single time point, a single sample, but you have the full gamut of phenotypes from the epithelial to the mesenchymal. Plotted again, now this is not VSNI, this is just simple expression of E-cadherin and vimentin. We can actually see the developmental progression of this transition, and we developed another algorithm that allows you to actually walk through the data in high dimension and find developmental progression. Again, we showed it in B cells that it's very accurate, very robust, very high time resolution. So now we can take every cell that we measured, each of our individual cells, and order them according to their developmental progression from epithelial to mesenchymal cells. And now that we've ordered the cells, we can actually look and see, how do the networks rewire? How do the interactions between different regulatory proteins change over time? And we can look on this sort of time axis that we've inferred from the data from a single time point and try and see how do these relationships change. Again, looking at the data here, for example, you could see how ERK and how MEK and P38 influence GSK3B. So this is basically a heat map of, based on the expression of P38 and PMEK, how is GSK3B influenced in height here? And you get these relationships. Of course, you could see that clearly these influence it. And then we can ask very naively, let's look at the beginning and let's look at the end. And what are some of the relationships? What are some of the influences of one phosphoprotein on another that change most dramatically during the course of this transition? And we see here this big line focusing on GSK3B, which is really a central hub in this network. And we see that there are many signals that are driving EMT that are very strong initially, but change in their strengths as EMT completes. And let me show you again a few data examples. We're now, one of the axes, rather than looking at an interaction between two proteins in a third, we're going to do EMT time here versus PMEK and see in slices how this change. At the beginning, PMEK really has a big influence on GSK3B, which wanes over time. We see a similar situation with P38. And actually AKT, which at least in the literature I read, and I'm not a big expert here, I'll put that caveat out there, was supposed to be a major player here, didn't seem to have much information and drive the event much. And again, we can actually look at temporal patterns. Again, we're driving this temporal dynamics from a single sample. We're not taking a time series. And we see first the influence, and here this y-axis is how strong the influence between two phosphoproteins changes. The influence of PMEK on GSK3B peaks right here, and after it, downstream beta-catenin peaks on its influence on ZEB and SNAIL, two transcription factors that drive this process. So using this time snap data that we got from a single snapshot, we can predict that maybe PMEK is important in driving this process. And another prediction is AKT might be less important or less effective. And indeed, here is again this sort of map from epithelial to mesenchymal, now in a heat map. Untreated, nothing much happens. Treated with TGF-beta, you see the mesenchymal phenotype emerge in the cell line. When you inhibit MEK, it doesn't die down completely, but you could see it's significantly removed. You see the percentages down here. And with AKT inhibition, it almost seems to exceed what you get without AKT inhibition. By the way, we get very similar behavior with P38 right here. So again, it correctly predicted MEK as a major driver of this process. And this is still very preliminary work, just to show you the power of what you can do once you measure things in so many dimensions. So finally, acknowledgements. I'd like to acknowledge, first of all, my co-conspirator, Gary, for the majority of the technology, many of the algorithms, and of course the wonderful data on AML. I then worked with Bernd, who was one of his ex-postdocs on the EMT stories, and most of it for my lab was led by Jacob, the main AML story, and Erin and Sean in Gary's lab, and the final story is Smita and Nirvana. Thank you very much.